So, welcome. My name is Marc Vaisband, and today I will talk about the validation of genetic variants from NGS data using deep convolutional neural networks. And I'll give a brief introduction to the topic, and I will then point your attention to particular detail, and lastly present the solution we have for this task. So, for the general topic, we are in the typical setting of wanting to identify somatic variants in a cancer using a sample pair of a tumor sample and a reference germline or a normal sample. And so typically this would look like the follows. We would sequence the data. We would do some automated QC. We would map the reference genome, use a variant caller to get candidate variants, annotate them using some database, and lastly, refine them manually to weed out sequencing artifacts or misalignments which were called as variants. And so the goal is to automate this step because, as you can imagine, as is, it's a bottleneck time-wise and also not very reproducible. And so we'll see this indeed possible. And so, for example, this would be a typical mutation. Right, so we have a clean sequence context, nothing to concern us. And then on the other end of the spectrum, there are also points like this one, which are also called by the variant caller. Right, even though as a human, we see that this is just sequencing gone horribly wrong, and there is no information here, it's just garbage. And so between these 2 points, there's of course many gray zones. But I'd like to look at the following. And this is the detail I want to point out. We have here a position. If this was live, I'd love to do a poll. He would say that this is an artifact as opposed to a mutation. Because really this is quite reasonable at first glance until we start opening the same position in other tracks which were sequenced on the same library as the tumor, because here it's important the germline and the tumor were sequenced on separate libraries. And so if we take just some random file from the tumor library, we'll be able to find the same deviation, oddly enough. We can keep opening more, and we always see the same picture. All the different tracks which we open which are from the same library actually exhibit the same deviation. And so we could think maybe this is some kind of recurring CLL thing. However, it also crops up in the germlines. So all of these are germline samples and these are CLL samples, which are completely unrelated to our sample pair, except for having been sequenced on the same library. And so if we summarize this, we see that we have 2 libraries, one of which has a significantly higher disagreement rate with the reference genome at this position. And so it just so happened that we had a germline sequence on one and a tumor on the other, and which then the variant caller looked like a genuine variant. And so the last point to address here is this data was obtained from transplantation experiments with mice. So one could think that maybe on library 2, we just had lines which exhibited this mutation more. However, that's also not the case. So if we deaggregate by the transplantation line, then we see that still the difference persists. It's really driven by the library. And so this is the main conclusion of our middle part. We really need this context to classify this kind of point. And this brings the last part, which is the machine learning solution to all of this. So we use neural networks because humans classify these points after a while using some kind of intuition, and these models are typically good at that. And we specifically use convolutional networks because they can leverage spatial information, which for us is important. And they have been quite popular recently in many image processing tasks. And what we have to begin with is the manual notation of 2082 called candidate SNPs. And what we do is we encode them into a numerical form, because obviously we need that to do some kind of machine learning. And for that, we take each track and tensorize it into a 3D object where we have one axis corresponding to position along the genome, so along the DNA, and one axis for the reads. And then lastly, in the depth dimension, we encode information such as the nucleotide and quality scores. And then once we have that, we can take the germline, the tumor, and then two random context tracks which are unrelated but from the tumor library, and then stack them along the depth dimension, yielding one data point. And so with these data points, we train a model which has a relatively straightforward architecture. So this is quite well known from image processing. We take blocks of convolutions and then follow up a max pooling just to extract high-level features, and then what comes out is fed into a dense classification head. And so this brings the results for which we do 5-fold cross-validation, and also to show it was not just a lucky seed for the cross-validation, we do just 100 random trained test splits, and every time we have this holdout set, which is not involved in the training, and on which we then evaluate the model. And these are the scores it yields. So we get a 97% accuracy, which is in line with the trained human researcher following published procedure. And so with this, I am at the end of my talk, and I will be very happy to answer any questions.